So pulmonary veins are coming into the left side of the heart from the lung, and into the left atrium, and being pumped out of the heart by the left ventricle. So what additional structures could we add to that that would have oxygenated blood? The aorta, okay? And then the pulmonary trunk, superior vena cava, inferior vena cava, coronary sinus, right atrium, left atrium, would all have deoxygenated blood. The anterior interventricular artery is associated with, you can find it on the heart, with which of the following? The great cardiac vein. Okay. What would we find with the small cardiac vein? Two arteries. The small cardiac vein is found with two arteries. Marginal artery. No. Marginal artery, and then it turns and goes around the belt of the heart, and the atrioventricular sulcus is what? So we have coming off of the aorta, the right coronary artery. It gets off the marginal artery branch, and we find the small cardiac vein with that. No. And then the right coronary artery continues around, and so the small cardiac vein continues around from front to back with the right coronary artery. So marginal artery, and then it turns and travels with right coronary artery, and empties into the coronary sinus, while the right coronary artery gives off the posterior interventricular artery. So it's marginal artery and right coronary artery. Middle cardiac vein is found with the posterior interventricular artery. Okay? And the great cardiac vein is found with two. In the front, circumflex artery, and then anterior interventricular artery. Okay? So now as we're thinking about that, hopefully you weren't seeing the words written out and paired, but you were visualizing the images of the heart, okay, and how they travel around. Okay, cardiac muscle is similar in appearance or structure to smooth muscle or skeletal muscle. Skeletal muscle. We spend a lot of time on, we skipped over that actually, in my 430 class. Innervation is similar to smooth muscle. Cardiac muscle and skeletal muscle are both innervated by the autonomic system. But structure-wise, it's similar to skeletal muscle in the sense that it is striated. What gave the appearance of striations in skeletal muscle? Right? The Z-band, the whole structure, what do we call the Z to Z structure? The sarcomere. Okay? And the sarcomere was formed of overlapping actin and myosin. And that gave us our anisotropic and isotropic, our A and our I bands. And so we have the same pattern. We have Z discs, and we have A bands and I bands. So we have overlapping actin and myosin forming the structure, the organized structure of cardiac muscle. There's some small differences as far as where, remember we had the triad, where we had the transverse tubule, and a dilated terminal cisternae of the sarcoplasmic reticulum on each side. So here would be the transverse tubule, and then the sarcoplasmic reticulum or the terminal cisternae on each side. In the skeletal muscle and cardiac muscle, just one. We have a dyad. So there's some small minor differences like that, whether this is associated with the Z disc or with the I band type of thing. There are some differences, and I have those listed on page 30. But this is contractile muscle. We have two different types of cardiac muscle cells. And this is what the contractile muscle cells look like. Okay? A difference between both skeletal and smooth muscle is that cardiac muscle, contractile muscle cells are branched. So you may have, here's one cell, and it's divided into two branches. And where these cells attach to another cardiac muscle cell, we have the presence of junctions. And because there's a lot of overlapping folding of the cell membrane, those membranes become visible as dark lines, and the histology of the muscle tissue. You can see them here. And those are identified as intercalated discs. Smooth muscle and skeletal muscle don't have those. Skeletal muscle runs the length of the muscle organ, so it's not forming a junction with another muscle. What the intercalated disc, as we'll see when we get to contractile cells, have gap junctions. And that allows a current to flow through muscle cell to muscle cell. So we don't have every single muscle cell innervated by the autonomic nervous system, as we do with skeletal muscle cell. Each skeletal muscle cell has a synapse with the bouton of a motor neuron, a lower motor neuron. That's not the case with cardiac muscle cells. And so there's a wave of depolarization that occurs downward in the atria, upward in the ventricles, and this wave is due to the gap junctions that allow that current to flow through. So here is a larger view of the cardiac muscle cells. You can see that there's only a single nucleus, similar to smooth muscle cells, but instead of being corkscrew shaped, it's sometimes described as a boxcar, because it's a large, almost rectangular nucleus. And a lot of mitochondria, because of the amount of energy that these cells require. Cardiac muscle cells will use glycogen, carbohydrates, but also lipids. All right, unlike our skeletal muscle cells, they have the capability of using fatty acids for energy as well. So here's a view of the intercalated disc. Desmosomes are found on the vertical aspect of the intercalated disc. So the intercalated disc is going to look like this. And these structures here are going to be desmosomes. Those are the spot junctions. Remember, they have the desmosomes have that plaque in the center, and then within the cell they have intermediate filaments that help resolve these with epithelial cells. So you can imagine if we have cell-to-cell junctions and they're pulling on each other, the tendency would be to pull those membranes apart. So desmosomes help to hold the cells together, and then on the horizontal aspect of the fold, we have the gap junctions that allow the signal, the depolarization signal, to travel from one cell to the next. Let's do a little bit of review. Favorite subject from last semester? Action potentials and potentials in general. So at the point where the arrow is showing, how do we identify that change in the membrane positive and negative nature? Depolarization. Depolarization. Okay. Now, the previous event, so what we saw as depolarization, would most likely be due to which of those events? A. Inflow of sodium ions. Okay. So let's do a little bit of a review of membrane potentials. If this seems unclear to you or you've forgotten some of it, you can always go back to the chapter on membrane potentials with the cell, or the review of that with nerve and muscle tissue. So a resting membrane potential the difference between the number of positive ions just inside the membrane and just outside the membrane, and we talked about it with the skeletal muscle cell or with the neuron, was due to the most part to the presence of three components or characteristics of the membrane. You guys remember what that was? What three characteristics of a skeletal muscle cell or a neuron contributed to a negative resting membrane potential? What? One at a time.
The leaky channel?
Okay, so we had leak channels, leaky channels.
Sodium pump.
So we have the presence of leak channels or leaky channels. And there were more potassium channels than sodium channels. So that meant that we lost more positive ions than we gained, right? Due to greater diffusion of potassium. So potassium is going out of the cell because it's in higher concentration outside the cell than it is inside the cell. Potassium is diffusing in, but not as rapidly as potassium is diffusing out. Okay? So that was one reason. What was another reason? Still talking about sodium and potassium. Okay, save that for the third reason. But still thinking about potassium and sodium. What was this another cause?
Sodium potassium pump.
Yes. So we have the sodium potassium pump. So using energy, free sodiums are pumped out for every 2 potassiums that are pumped in against their gradient. Again, a greater loss of positive ions. Okay, and now the protein. There are large proteins which cannot cross the plasma membrane inside the cell that have a net negative charge. So this gave us a net effect of more negative ions or fewer positive ions just inside the plasma membrane than outside. Okay? So leaky channels, more potassium channels than sodium. Sodium potassium pumps, so more sodium is pumped out than potassium is pumped in. And then the negative charges of the protein. Now, after we talked about that, we talked about the different types of channels that cells have. Or maybe we talked about that first. I don't remember. Did I erase this? You guys still looking down? Okay. Of all of them, the potassium leak channels, so we had the leak channel, which meant they were constantly open. Okay, they didn't have gates. And we have them primarily for sodium and potassium, and so we said we had more potassium leak channels than we have for sodium. And voltage gated channels. What were voltage gated channels? All right, so as a current, how do we define a current? I skipped the chemistry lecture last semester, but how do we define electrical current? What was it? The flow of charged particles, all right? So if we have a membrane with a protein channel in it, and a flow of charged particles, which we're talking about ions here, goes by, if that is of insufficient strength, not enough ions, nothing happens to the shape of this channel. However, if a greater number of charged particles flows by, it causes the channel to change shape, and the gate moves, and the channel is now open. So it could be open for sodium, which will allow sodium to come into the cell. It can be open for potassium, which would allow potassium ions to leave the cell. It can be open for calcium. Calcium ions would come into the cell. Those are the three major types of voltage-gated channels that we have. Do you remember where we had the voltage-gated calcium channels? Where did we see voltage? The synaptic bouton. Where did we have them in skeletal muscle? In the transverse tubules, just inside the transverse tubules. Sarcoplasmic reticulum. Because that was where we had our storage for calcium ions. Okay? So as the action potential traveled deep inside the muscle cell along the transverse tubule, that current would open the voltage-gated channels of the sarcoplasmic reticulum, releasing calcium ions for the muscle cell. All right? Anybody remember the third type? Okay, ligand or chemically gated channels. So obviously when we're talking about neurons, it would be any of our neurotransmitters. Okay, so dopamine, acetylcholine, norepinephrine, serotonin, any of those neurotransmitters. When we talked about skeletal muscle, what was the ligand that opened the voltage-gated channels at the motor end plate? Acetylcholine. So we'll look at how we modulate the heart's activity with ligand-gated channels. And those ligand-gated channels in the heart will be for the sympathetic and parasympathetic neurotransmitters, for acetylcholine and norepinephrine, epinephrine. And then we talked about mechanically gated channels, which we don't see effective in the heart, okay? But pressure, or the sound waves, or fluid waves can activate mechanically gated channels. All right, so a review of channels. And now let's review potentials. So the potential is the difference in the number of charges just inside the cell membrane and just outside the cell membrane. Okay? So we talked about the resting membrane potential already. In the neuron or the muscle cell, when we opened a ligand-gated channel, what kind of a membrane potential did we create? A ligand-gated channel or a mechanically gated channel? A graded potential. So remember, depending on whether we had a lot of neurotransmitter present, we might transiently open it like that, or a smaller one, or a larger one, depending how many ligands, how many neurotransmitters were binding to the ligand-gated channel. So graded means different degrees of current are produced. So graded potentials are associated with ligand-gated channels. So in the neuron, where did we find these graded potentials being produced primarily? You thought you were all done with this stuff. So when we activated a neuron, did we activate it directly into an action potential, or do we produce a graded potential first, typically? A graded potential first. And that graded potential may inhibit, make it more difficult to create an action potential, or it might make it easier. We talked about EPSPs and IPSPs. So where were those formed? On the axon or on the cell body? Cell body or dendrites. Okay? In skeletal muscle, where did we create graded potentials? What did we call the region of the synapse of a lower motor neuron and a skeletal muscle membrane? The motor end plate. So remember the diagram here. We had amniotic gluton coming in like this. And these channels for acetylcholine were found in the motor endplate. And if sufficient numbers of them were opened, then an action potential, a greater potential would be strong enough to open our voltage-gated channels along the rest of the plasma membrane. So the next type of potential would be a threshold potential, which was the minimum graded potential sufficient to create and to open our voltage-gated channels. And then it could also be, it could be created by either another action potential or by a graded potential. But it was the minimum current required to open a voltage-gated channel. And then once we reach that, an action potential is created. And action potentials were created, well, that was an all or none response. And those were associated with voltage-gated channels. Hopefully, these concepts are coming back a bit. So now if I were to diagram action potential of a skeletal muscle cell, let's place these concepts on that diagram. All right? Can I erase the board? Can I erase the board? You guys have that down?
1 second.
Pardon?
One
Okay.
second.